Welcome to DNA replication, transcription, and translation. Bodies of all animals and plants are made up of tiny microscopic unit called cells. In the cells, we have nucleus that have chromosomes codified in it. They have genes located in them and are responsible for storage of genetic information in a coded manner. The coded genetic information is stored in a twisted double-stranded molecule called DNA. DNA replication. During DNA replication, the following steps takes place. Step 1. Untwist. The two twisted strands forming the double helix and wings. Step 2. To unzip. The two now untwisted strands forming double helix separate. That is like doing a zipper. This is made possible because of the presence of weak bonds that link the bases of two strands. This process is under the action of replicating enzymes known as helicase and result in the formation of replicating fork. The separated strands now act as a template for formation of a new strand. Step 3. Copying DNA to a new strand. The information of the DNA in form of a base sequence is copied onto a new but complementary strand using the parent DNA template. This is made possible by ensuring only the complementary bases link up with those on the template strand, such that thymine would only pair with the adenine and guanine would only pair with the cytosine when forming a new DNA strand. Step 5. Sealing. Covalent bonds are then formed to seal between the new and the old strand, resulting in the formation of a new DNA strand. Note that both the template and the new strand then undergoes coiling to form a double helix. By so doing, DNA is said to have doubled itself so that sufficient DNA are made available for each of the daughter cells during cell division or during repair of worn-out tissue. Welcome to DNA transcription. DNA transcription means formation of a messenger molecule called messenger RNA that is formed from DNA strands. Messenger RNA is able to leave the nucleus with full instruction from the DNA strand as a coded information for the synthesis of a specific amino acid. DNA transcription begins by step 1. Double helix of the DNA unzips and free nitrogen bases align themselves opposite the parent template under the action of enzyme called RNA polymerase. Step 2. The base sequence of the template is copied onto a new strand, which then becomes an RNA strand in the same manner as during DNA replication. However, unlike DNA, in the formation of messenger RNA, nitrogen-base thymine is replaced by the base uracil that gives it ability to leave the nucleus. When the transfer of DNA base sequence onto the messenger RNA is complete, the messenger RNA leaves the nucleus into the cytoplasm with the full instruction from the DNA about the kind of protein to be synthesized by the cell. Welcome to DNA translation. Information on messenger RNA is translated by the ribosomes and is used to assemble amino acids into specific protein molecules. This is normally done by another type of RNA molecule called transfer RNA. Transfer RNA reads 3 bases at a time, forming amino acids that are continuously added to form a long chain of amino acids. And once this is done, transfer RNA molecules transport amino acids from their pole in the cytoplasm to the protein assembly point. This is followed by condensing of amino acids to form a long polypeptide chain, which is the protein. Protein molecules then determine the inherent trait in organisms. Welcome to detailed explanation of DNA transcription and translation. During protein synthesis by the ribosomes, 2 processes are involved, that is, DNA transcription and DNA translation. DNA transcription means taking a single gene of DNA and copying it to messenger RNA, whereas translation means taking this messenger RNA strand and using it to make or produce protein. Why transcription and why translation? In order to make a protein, a sequence of nitrogenous base has to be read by ribosomes that are found in the cytoplasm of the nucleus. In our previous explanation, because DNA is so long, it can't leave the nucleus. This means if we want to use a gene to make a protein, we need to make a copy of it first. And because it's a single gene and not the entire DNA strands, the copy will be small to live through the small nuclear pores and reach the ribosomes. This copy is what we are calling messenger RNA. Messenger RNA is able to leave the nucleus because of the following 3 reasons. 1. Messenger RNA is just a copy of a single gene and is similar to DNA. However, it's shorter than DNA because it's just a copy of a single gene and not the entire DNA strand. And 2, it's a single strand and not a double strand like DNA. Lastly, instead of containing base thymine, it contains base uracil. This is how messenger RNA is formed. That is transcription. The double helix of DNA uncoils, showing the bases: adenine, thymine, guanine, and cytosine. Suppose we want to make the highlighted gene. We start with enzyme RNA polymerase. The DNA strands are separated like doing a zipper. The enzyme RNA polymerase moves along the DNA parent strand, reading it, and forming the messenger RNA strand that is complementary to DNA strand, just like during DNA replication, such that adenine pairs with thymine, guanine pairs with cytosine. However, when we have adenine in DNA strand, it will pair with an uracil on messenger RNA. Take a look. Once done copying the entire gene, the messenger RNA strand detaches and is free to leave the nucleus heading to the ribosomes. When the messenger RNA leaves the cell nucleus, it gets attached to the ribosomes that translates the instruction in messenger RNA, a process known as translation. Note, whether it's DNA or messenger RNA, each group of 3 bases codes for a specific amino acids. To make protein, our cells uses 20 amino acids, and each of them has 3 basic codons. For example, NGU codes for amino acid serine, while CCA codes for proline. These are just examples, you don't have to memorize them. The translation. This begins with our messenger RNA attaching itself to the ribosomes and starts adding 1 amino acid at a time. The codons, which are the amino acid, are pulled by another type of RNA called transfer RNA. Transfer RNA has a specific amino acid with an anticodon molecule that must attach to a complementary code on messenger RNA molecule. Because each transfer RNA molecule carries a specific amino acid, our UCA, for example, will be attracted to AGU on the messenger RNA molecule because they are complementary to each other. Same case happens to transfer RNA anticodon GGU will get attracted to CCA, and by so doing, T transfer RNA has corrected amino acids in their correct order, so the ribosome is able to join them together to make a chain of amino acid, and by moving along the strand of messenger RNA, where the formed amino acids are condensed to form a long polypeptide, which is a protein. And this protein now is responsible to make a new cell during cell division and also to repair any worn out tissue or formation of a new organ.